Hello everyone, welcome to Biotechnica. So today I have come up with yet another important topic, which is oogenesis. So you'll see the same in developmental biology, as well as you'll see the same oogenesis in case of system physiology in animals. So this is one of the important topic that we're going to talk through the mind maps. So let's dive into the session and understand what it is all about. So first, let's understand what is oogenesis. Oogenesis is a process in which a female gamete or ovum or egg is actually produced. This usually takes place in a female reproductive system. So let's talk about when will the process of oogenesis begins in case of a females. So if you talk about in case of man or boy, you can literally see there is actually a continuous production of sperm. But in case of women, there would also be a discontinuous process that you can actually see. When a woman, or we can say a female embryo is formed, this female embryo, in the embryo itself, there is actually a production of something called as oogonia, or we can call it oogonium. So you literally see in the embryonal stages itself, there are going to be 7 million oogonium present in the ovary. So in the ovary, you can see literally, if you go and examine the female embryo, in the ovary, you literally can see 7 million oogonias are present. Later, this 7 million oogonia will undergo atresia, or it just degenerate, and literally you can find 2 million oogonium or oogonia present in the female ovary, very specifically in the embryonal stages itself. This oogonia is now 2 million and no more oogonia will be added after that moment. So this is in the embryonal stage. After, or we can say before the puberty, whatever it is happening some 14 years before or 15 years before. So pre-puberty time, so what exactly happens? This oogonia is going to divide. So I'm writing only one very specifically. So every oogonia is going to divide. So I'm writing oogonium is going to divide and it undergoes mitosis, and you're going to see primary oocyte. Primary oocyte. And this primary oocyte, it's a cell actually, which is a diploid cell. And this diploid cell will be surrounded by a lot of granulosa cells. So this is going to be the granulosa cells. And this is the primary oocyte, or we can say this is the primary oocyte. So the primary oocyte is surrounded by a lot of granular cells. Now I'm going to call it as primary follicles. So you should know the difference between a primary follicle and a primary oocyte. So we can say a primary follicle actually contains a primary oocyte present inside it. And this primary oocyte is definitely going to be a diploid cell. So we can say the primary follicle or the primary oocyte will be halt, or it will be just stopped. It will not undergo meiosis. So it just starts with meiosis 1. And in the meiosis, very specifically in the prophase 1, it will be halted. Only at the time of puberty, this primary oocyte will go in for meiosis 1 complete division. So still now we have seen in a female ovary, we can see at the time of puberty, how many follicles are present? Almost 40 to 70,000 follicles are actually present in the female ovary at the time of puberty. So let's understand the points. So the process of oogenesis begins in the fetal stage itself. Yes, we know in the embryonal stage itself it has started almost 7 million, and then it's reduced to 2 millions, and after that 2 millions is reduced into 40 to 70,000 primary follicle before the puberty itself. So when a large number of primordial cells in this term is referring to the primary follicles, which contains primary oocyte, which is diploid. Everything is formed by mitosis, yes. A oogonium undergoing a mitosis and produces lot of primary follicles, yes. And where is the location of this one? So if I have to talk about the ovary, we know there are going to be pair of ovaries which are actually present, and from here you will see the fallopian tube actually going like this, and then you see the uterus part, and the vaginal part like this. So where is this actually taking place? Yes, exactly in the fetal ovary it is actually taking place, but in the epithelium, germinal epithelium. And these germinal epithelium are made up of germ cells, and one of the germ cell is going to be the oogonia. So almost you have 2 million oogonia, and all the 2 million oogonia will undergo division and they form lot of primary follicles. We can call it primordial cells, and these primary follicles are going to have primary oocyte, and this will be halted before the puberty. And at the time of puberty, this primary oocyte will undergo meiosis. So after puberty, every month there is a production of one egg. Yes, very specifically, this is not egg, this is actually a secondary oocyte. A secondary oocyte will be coming out, and 3 polar body, this is actually variable, 2 to 3 polar bodies will be found. And this germinal epithelial cells in fetal ovary functions as germ cells. I told you these are germinal epithelial cells which are going to act as germ cells, or we can call it oogonia. Later these germ cells undergoes meiosis 1 and meiosis 2 and forms a secondary oocyte. And this secondary oocyte, when they encounter a sperm, then they will be making a ovum. Without the entry of the sperm towards the fallopian region, this secondary oocyte will not undergo meiosis 2. So then we will actually get an ovum, and then the sperm comes and fuses together very specifically at the ampulla part, at this region of the fallopian tube. Fallopian tube, we already know it's going to be the oviduct. Okay. Now, what are the stages that you see in case of oogenesis? There are 3 stages, which is going to be the multiplication phase. Very simple, where oogonium is going to multiply rapidly, and in the growth phase you have lot of primary follicles which are going to grow, and in the maturation phase you will be seeing a primary oocyte is going to be converted into a secondary oocyte. And if a sperm encounters, then it's going to make a ovum. So we are going to see 3 important phases in case of oogenesis. So now let's understand what are the phases in detail. Everything will go in detail of the phases. Now let's understand what is multiplication phase. So in multiplication phase, the germ cells which are present in the fetal ovary is going to divide by mitosis. So how it actually happens? So let's understand. So first they are going to have, let's suppose take this is the ovary. So in the ovary, what is going to happen? So in the ovary, I'm going to write the germ cell in the fetal ovary divides by mitosis to form 7 million egg mother cells or oogonia. So I'm just telling that there are a lot of germinal epithelium inside it, and we're going to take only one germ cell, and this germ cell is going to be diploid in nature, and this germ cell divides by mitosis. Germ cells divides by mitosis, and you're going to get almost 7 million oogonia. So 7 million oogonia are actually present in the embryonal stage itself. Later what's going to happen? So this is a normal mitosis process. So in the fetal ovary, you're going to have a germinal epithelium where the germ cell is going to undergo mitosis to form 7 million oogonia. And these oogonia multiplies. Yes, this also multiplies. And some die, most of them die by apoptosis. That's why I told almost 2 million oogonia only will be seen in the embryonal stage of the females or female embryo. So because most of them will die at this stage, so that only 1 to 2 million oogonia will be present at the time of birth. It's not more than that. Now, what happened to the oogonia? So let's consider this is the oogonia which is actually diploid. Now, all the oogonia are formed in the fetal stage and no more addition. So, only till 1 to 2 million oogonia will be seen in the fetal stage. And after birth, no more oogonia will be added. The next, the oogonia forms egg tube into the stroma of ovary, thereby forming a lot of egg nest. So, what is exactly going to happen? This oogonia is going to divide by mitosis, and then they are going to form a primary oocyte. So I'm going to call it as primary oocyte. this primary oocyte is also going to be diploid in nature. You have to remember this is the primary oocyte which is diploid in nature, and now they will not go for meiosis. This is all happening before the puberty, not after the puberty. All are happening before the puberty. So now let's understand about the growth phase. So now these cells, which we call it oogonia, undergo cell growth. Yes, now they have undergone a growth, and then they are large enough to undergo meiosis 1 to become a primary oocytes. So we are just telling this primary oocyte is going to be formed, but still now we are not calling it anyway. So which means the meiosis 1 has started. The meiosis 1 has started, but it is not completed. We know meiosis 1 and meiosis 2 has to take place. So we have the primary oocytes. And now this primary oocyte will be halted before the puberty. It will be halted before the puberty. At which stage? The primary oocytes are arrested in prophase 1 of meiosis 1. Very specifically prophase 1 of the meiosis 1, it will be arrested. And this primary oocyte, I'm just drawing, this is the primary oocyte. Now it will be surrounded by a lot of granulosa cells, or we can say columnar and cuboidal cells. So this is going to be the primary follicle. So you can see the primary oocytes are arrested at prophase 1 and surrounded by granulosa cells. And now I'm not going to call it primary oocytes. I'm calling it a primary follicles. So what you have, a primary follicle, and this primary follicle is going to have a primary oocyte. Primary oocyte now. And the primary oocytes remains arrested in prophase 1 of meiosis 1 until the puberty. After the puberty only this meiosis 1 will be completed. Until that it will just be arrested at prophase 1 of the meiosis 1. Now a large number of primary follicles undergoes. I told you almost lakhs of primary follicles will be formed, because you already seen 2 million oogonia are present, and out of that almost lakhs of primary follicles will be formed. Out of that almost 40,000 to 70,000 primary follicles only will be seen at the time of puberty, which means all of them is actually going to be, which will undergo atresia, or we can say degradation process. So now in the growth phase we have seen the primary follicles. So the primary follicle is present, which is going to have a primary oocyte, and it is seen in the growth phase. So I'm writing growth phase as GP. Now each month the women is going to have the hormone stimulation, which is going to be FSH. Now the question for us, where is this FSH coming? FSH actually comes from the hypothalamus. Actually, if we have to talk about that, the hypothalamus is going to produce gonadotrophin releasing hormone, and this gonadotrophin releasing hormones goes to the anterior part of the pituitary, and now the pituitary is going to secrete follicle stimulating hormone and luteinizing hormone. Now, this follicle stimulating hormone is going to trigger, this primary follicle is going to have a primary oocyte. So, the concept is this primary oocyte is going to undergo meiosis, meiosis 1, because previously before the puberty it is just halted. And now during at the time of puberty, what exactly happens? Meiosis 1 is going to take place. This is diploid. And now meiosis, what will happen? One haploid set will be formed, and this is going to be the polar body. This polar body will have a less amount of cytoplasm, whereas the other one is going to have more amount of cytoplasm. And now we are going to call it as what? Secondary oocyte. And now this meiosis 2 will take place. The next step, the meiosis 2 will take place only when the sperm encounters. So then this secondary oocyte will undergo meiosis 2 and it forms a ovum and the second polar body. But this step, this meiosis 2 will happen only when the sperm encounters, or else still meiosis only will happen. So during our ovulation process, the secondary oocyte only will come out of the body. This secondary oocyte will be absorbed or taken by the infundibulum of the fallopian tube. And this secondary oocyte will be present in the ampulla region of the fallopian tube. Only when the sperm comes, this secondary oocyte will go in for meiosis 2 to form the ovum, and and then fuses with the sperm and makes for fertilization process. So now let's understand the points which is given. So each month a woman follicle stimulating hormone will triggers the continuous division of the primary oocyte. Okay, we already know the structure of a primary oocyte. So this is going to be the primary oocyte, and this primary oocyte is surrounded by lot of Graafian follicles, and now we are calling it as primary follicles. So as the follicle stimulating hormone comes along with the luteinizing hormone, follicle stimulating hormone is actually helpful for the follicular development or division by converting the cholesterol into androgens, and luteinizing hormone is the one which actually produce or estrogens, we can say. Now this primary follicle, what it will happen is it will go for further division. So now this primary oocyte is still present. It is diploid in nature. And now this is surrounded by yet another layer. And this layer is called as zona pellucida. This is zona pellucida. And they are actually surrounded by lot of granulosa cells. This is called as the early primary follicles, early secondary follicle. And later, even more amount of follicle stimulating hormones will come. Luteinizing hormones will come. That time there would be maximum production of estrogen. Yes, even the cell divides further. So now you have the primary oocyte, and then they are surrounded by zona pellucida, and many more granulosa cells will come and bind together. And now I am calling it late secondary follicles. And this late secondary follicle will have some spaces, and these spaces are actually filled with hyaluronic acids. So we can say follicle stimulating hormone will do this action, and there will be some fluids filled spaces. And now this late secondary follicles will then convert into a Graafian follicle or a tertiary follicle or a mature follicle. What's the difference of this? So here you will be having very important thing. Till now, you have seen only primary follicles, and only in the Graafian follicle you will see the secondary oocyte, which means the late secondary phase, later it undergoes meiosis 1 to form this. And now I am not calling it primary oocyte, I am calling it secondary oocyte. And now the secondary oocyte is haploid. It's not diploid, which means only when a Graafian follicle is formed, the meiosis 1 is completed. And they also have a zona pellucida, and they are surrounded by lot of Graafian follicles, and corona radiata, everything is present, and they will make an anthem, a lot of space here. So they'll make a big anthem. So you will see anthem formation only in the Graafian follicle, which means this Graafian follicle is going to have a secondary oocyte. So what has happened? From a primary oocyte has undergone meiosis 1, and it has actually formed a secondary oocyte which is actually haploid. This is diploid. This is haploid, which is secondary oocyte. You will see secondary oocytes only will be released every month. This secondary oocytes will be taken from the ovary into the oviduct. And only when the sperm encounters the secondary oocytes will undergo meiosis 2 and produces ovum plus another polar body. Now let's understand these points. So these cells undergoes first meiotic division to form two cells of unequal size, which means a primary follicle containing a primary oocyte, which is actually going to be a diploid one, and they will undergo meiosis 1 only. And now they are going to form a secondary oocyte. This is haploid. A secondary oocyte, which is a haploid, and a small polar body, primary first polar body. This is a first polar body. So it forms two cells of unequal size because this secondary oocyte is going to have lot of cytoplasm, whereas the first polar body is going to have less amount of it. One cell retains the entire cytoplasm to form a secondary oocyte. Yes, we have drawn a secondary oocyte which is haploid. And the other cell form a polar body with less cytoplasm. The polar body will remain in the primary follicle itself. Still now, it is actually present in the primary follicle, and then it is going to present in the secondary follicle. The secondary oocyte will be taken by the fallopian tube. So this is the ovary, and here you are seeing the fallopian tube. So now what we have the secondary oocyte. And now the secondary oocyte will be taken into the fallopian tube, and only when the sperm encounters, until that the secondary oocyte, we can say it's a haploid secondary oocyte, it will be halted. It will not undergo meiosis 2. It will be halted. And at the phase of metaphase 2 of meiosis 2. Only when the sperm comes, then it will undergo meiosis 2. So the secondary oocyte is released from the ovary every month. So only the secondary oocytes will be coming out every month. If a sperm enters, then the fertilization will actually take place. So meiosis 2 is completed after the entry of the sperm into the fallopian tube. Suppose the sperm comes here. Then the secondary oocytes will undergo meiosis 2 and they will form ovum plus a second polar body, and this ovum will fuse with the sperm in the ampulla regions. Yes. So now we have understood the maturation phase, and now let's talk about ovulation. What is ovulation? We know that the Graafian follicle is the one which has the secondary oocytes, and the Graafian follicle is going to rupture and releases the secondary oocytes, and this secondary oocytes goes to the ampulla region of the fallopian tube, or it will be taken by the infundibulum through the fimbriae. And now, after the Graafian follicle ruptures, what will happen? The secondary oocyte will come as a menstrual cycle. So that process is called as ovulation. So you can see, at puberty, it is the release of the secondary oocyte and not the ovum every month from the Graafian follicle by any one of the ovary. It can be onto the left ovary or can be onto the right ovaries. The ruptured Graafian follicle, okay, the Graafian follicle has ruptured, and now this Graafian follicle is called as corpus luteum. And this corpus luteum is the one which actually going to release or secrete progesterone if the woman is actually fertilized. Only when the sperm comes, the secondary oocyte fuses with them and makes an ovum, and ovum and sperm will fuse together and a zygote is formed. And that time this corpus luteum is going to secrete lot of progesterone, which makes the uterine wall very, very thicker. And only 450 secondary oocytes are formed during the entire reproductive cycle of the females. And now let's see the image. So this is the entire process of oogenesis. So actually remember that the oogonia, or we can say the oocytes, are actually present in the follicles. So there is a difference of follicular sites and there is a difference of oogenesis. So you can see oogonium is present in the fetal stage itself, or we can say in the oogonia or in the embryonal stage itself. The second is primary oocyte arrested in the prophase before the puberty. At the time of puberty, this will undergo meiosis. So that time it will form a primary oocyte, and this primary oocytes are surrounded by a lot of Graafian follicle. We call it primary follicles. And later this primary oocytes will undergo meiosis 1 and then form a secondary oocyte. This secondary oocyte is actually present in the Graafian follicles. And during the menstrual cycle, this Graafian follicle break open and the secondary oocytes will be released. But if the sperm enters, then what exactly happened? This secondary oocyte will undergo meiosis 2 to produce ovum plus a second polar body. This ovum fuses with the sperm and makes up a zygote. Yes, now we have understood what is this. Now we have to understand the structure of an ovum or an egg. So you can see the structure of this. So this is going to be the egg or the ovum, which has a vitelline membrane. And then they have a zona pellucida, the second layer, and then they have a lot of corona radiata structures, and then they have this polar bodies present, and then they have the cytoplasmic content maximum present over there. So today what we have done is we have studied what is the process of oogenesis and what is the phases of oogenesis, which is the multiplication phase, growth phase and maturation phase. Then we studied about what is multiplication, growth phase and maturation phase, and we have also understood the process of oogenesis and the structure of ovum. So I believe that this video is helpful for you. If you really like this video, please like, share and subscribe to our channel Biotecnika. Thank you all of you for your time, and I'm going to meet you back again in the next video.